Hello, my name is Brooke Duke, and I was chosen to be a part of the Envision summer program for 2025. I am a junior pre-vet major with a minor in nutrition, and I've always had an extreme interest in research. This summer, I worked under Dr. Muyyarikkandy alongside his PhD student, UD Joseph, researching antimicrobial resistance. Antibiotic resistance is an increasingly relevant crisis across the globe. Due to overuse and improper use, antibiotics are becoming ineffective against many pathogens, like E. coli and Salmonella, which is resulting in a heightened cost of treatments, longer recovery rates, and higher mortality rates. In my project, I'm researching the effectiveness of the specific probiotic Enterococcus faecium, or MLS-15, when used alongside treatments that already exist against pathogens. There's a lot of preparation that goes into research, and I'll go over a few of my basic lab duties that I had to do every day to either maintain my bacteria or prepare for my experiments. I work with pathogens. Even my probiotic is considered a pathogen, so I must wear proper PPE at all times and always work under the fume hood. My project requires a lot of agar plates, so a lot of time and effort is spent preparing them. Not only do I have to subculture my probiotic, but I have to subculture my pathogens too. This is the backbone of my experiment. This makes sure they're all growing well and they're in a good condition to be tested on. You can also subculture by using a singular colony, which you can get by streaking your bacteria. MLS-15 was characterized genotypically using the Comprehensive Genome Analysis Service at Patrick, using BV-BRC, and phenotypically by assessing MLS-15's tolerance to lysosomes, acid, bile salt, gastric juices, and intestinal juices, as well as cell surface hydrophobicity, cell adhesion, hemolytic activity, antibacterial activity, and antimicrobial susceptibility. As you can see so far, there's a lot of different methods that go into these experiments. A lot of them use serial diluting and plating, which is used to enumerate the bacteria, so we can compare the control groups and the treatment groups. We streaked our bacteria on a blood-infused McConkey agar to see how it reacted with blood, along with a million other methods. And that brings us to our chicken embryo challenge. I assisted Jiddu in candling the eggs, marking the air sac, and many other things. This was actually on MLS-3, Jiddu's probiotic. We injected over 160 eggs with varying treatment and control groups. We then resealed them and let them incubate until the day of sampling. Here we sampled various organs through chicken necropsies for further testing. Samples were then assessed using either the qPCR, the Western blot machine, or through histopathology. I also attempted to see the effect of my probiotic MLS-15 on C. elegans. We bleached and hatched the worms successfully, as well as pre-treated the plates. We challenged the worms with treatments, controls, and combos of OP50, MLS-15, and Salmonella. We had about 15 plates between treatments and replicates, and I had to transfer 25 C. elegans into each one. This means there was about 375 worms that I had to keep track of and count every day, and transfer every other day. Until the fungus. Fungus was growing on my agar plates before I even put the worms into them. It wasn't long before we noted that the fungus acted as some sort of food source for the worms, and they reproduced exponentially. As you could probably assume, that messed up my entire experiment, and I had to stop. The purpose of these experiments I did with MLS-15 is to not only research antimicrobial effects, but to make sure it's safe for your body, and to make sure it can survive in your digestive tract, because it's no use if it's dead. I'd like to give a special thanks to the Envision program and all its supporting staff, as I've had such a fun summer learning and exploring my interests. I hope many students after me get to enjoy this awesome program as well.